Hi, my name is Matthew Gazzara. I'm a graduate student in the labs of Yosef Barash and Kristen Lynch, and today I'd like to talk to you about our analysis to identify novel regulators of 3' UTR isoform diversity. Cleavage and polyadenylation is a qPRAM RNA processing event carried out by core complexes. These core complexes recognize cis regulatory elements at the end of the pre-mRNA and dictate where cleavage occurs and the poly A tail is added. Importantly, the site of cleavage and polyadenylation is not constitutive, but alternative, leading to diversity in UTR sequences. So alternative polyadenylation, APA, is largely the result of choosing between competing polyadenylation sites or paths within a terminal exon that leads to shortening or lengthening of the UTR. Targeted 3' end sequencing techniques that directly determine the site of cleavage and polyadenylation across diverse organisms have shown that APA is widespread, with around 75% of genes in human undergoing APA. APA has important downstream consequences for gene expression, where we can add or remove binding sites for things like microRNAs or RNA binding proteins that go on and lead to altered mRNA stability and localization, for example. The field has observed a general association of 3' UTR shortening being associated with proliferative cell states, most notably cancer, while lengthening is associated with differentiated cell states. While this is an oversimplification and there are many exceptions to these rules, an outstanding question is, how is this APA achieved in condition-specific manners? The main working hypothesis in the field is that patterns are governed by the degree of expression of the core cleavage and polyadenylation machinery, where suboptimal proximal sites are filled as the core machinery's expression increases. And this is shown to be true for a select few cancers, but we believe that this is insufficient to explain the complex patterns of APA that you can observe across human tissues from GTEx from this study, when you compare that to expression levels of the core machinery, which are pretty high and ubiquitous. So much like the regulation of alternative splicing that's been well studied, we know that there are auxiliary RBPs that can sterically occlude or recruit core spliceosome components and influence splicing decisions. Similarly, in alternative polyadenylation, a handful of RBPs, like SELF2, studied in the Lynch lab, have been found to compete or recruit the CPA machinery to alternative paths within the same terminal exons, leading to differences in isoforms. Here I highlight a few such studies of individual RBPs that influence APA, but really we see this as a missing regulator problem, where there are many RBPs that exist within the human genome, but most have not explicitly been explored as potential regulators of APA. So our approach here is to utilize the compendium of RBP-related data from ENCODE to search for additional potential novel regulators of isoform diversity. Ideally, we'd have targeted 3' end sequencing data comparing to control to knockdown, but we do have a large set of over 500 RBP knockdown experiments with RNA-seq, and we can utilize that to identify APA targets. So we could detect APA directly from RNA-seq to quantify and identify regulators of APA patterns. You can see in this example, a number of tools utilize annotation and 3' end sequencing databases to define polyadenylation sites and measure quantifications of relative usage of those polyadenylation sites. And other tools still can detect these directly de novo, looking at RNA-seq coverage. So there are a lot of tools out there, and over the past year, myself and many others have done a community effort called APA Eval to evaluate these different methods and try and say which one's doing well. So there's a lot of information there. I encourage you to check out our talk and poster at this conference for more details. But my work here utilized the popular and, in our hands, well-validated tool DePARS that can detect de novo proximal poly-A sites, which we feel is crucial for identifying our previously unseen paths upon RBP knockdown. Applying DePARS to the large set of RNA-seq data from ENCODE, we find a number of APA regulators, and I'm going to walk you through a small subset first and then widen the scope. So here's a handful of RBP knockdown experiments, and we can see hundreds to over 1,000 genes with significant APA shifts using parameters, thresholds that we found in the Lynch lab to validate well by 3' race. Importantly, many of these are identified previously as 3' end processing factors, but many are not, so they're potential novel regulators. Next, we can look at the primary effect of RBP knockdown on the direction of APA change. So did the UTR get shorter when the RBP was knocked down, or did it get longer? And that's shown in blue and red respectively. Importantly, we see some positive controls in this initial analysis that are consistent with what is known in the literature. For example, we see that CSTF2T depletion leads to a 3' end lengthening upon knockdown. That's consistent with targeted 3' end sequencing experiments in cell lines that showed global 3' UTR lengthening upon depletion of this core cleavage and polyadenylation subunit. So showing half the data here, all the RBP knockdowns in one of the 2 cell types, HepG2, we see our positive controls right here. But the rest of the talk, I'm going to focus on DDX55, which is our number 2 candidate in HepG2 cells, which had the second most number of targets. And interestingly, it's a little hard to see, but almost all of the targets upon DDX55 knockdown were shortened upon knockdown. So why DDX55? When I initially started the project, there were actually no PubMed hits for DDX55. But being an RNA helicase from the dead box family, we know that many of these RBPs play important roles in RNA metabolism, in splicing, transport, ribosome biogenesis, decay, etc. DDX55 is in fact conserved down to yeast, where it's called SPB4. And in yeast, it's been shown to localize to the nucleolus, where it's involved in rRNA processing. But in humans, this protein has not really been well studied. DDX55 at the transcript level is broadly expressed in many human tissues shown by GTEx, but we do note there is some variability in expression levels, which may influence tissue-specific APA patterns. That's an area of active investigation. Additionally, it has been shown subcellularly to localize to the nucleoplasm, the nucleolus, and the cytoplasm in human cell lines. So it's in regions where APA could be happening, namely the nucleoplasm. If we look at DDX55 binding data, ECLIP peaks, we see that the binding is very specific and localized to 3' UTRs. I'm showing the fraction of all ECLIP peaks, and most of them are in 3' UTRs. Many transcripts do have DDX55 peaks in their 3' UTRs, where as many as 70% of expressed transcripts in both cell lines have some evidence of DDX55 binding in the 3' UTR. So next, we looked at the motifs that were enriched under DDX55 peaks, and they include sequences that look like binding sites for the core CPA machinery, namely the PAS hexamers found within the second motif, and UGUA, the upstream enhancing element, is also found as enriched. So it's not surprising when we look across experimentally determined polyadenylation sites from human liver that we see binding patterns like this, where DDX55, shown in green, is binding to many, many of these experimentally validated polyadenylation sites right near the cleavage site, in a manner that's comparable to other core CPA machinery binding events. So this gives us some confidence that this might be a direct regulation, not purely indirect. So now I'm going to combine our functional data from RNA-seq with our binding data to really interrogate that direct question. So if we look at a metaplot of all the 3' UTR shortening events upon DDX55 knockdown and compare the fraction of those events that are bound, that are regulated versus confidently non-changing, we see this metaplot around the regulated proximal and distal pass. And you can appreciate that there's enriched significant binding of DDX55 specifically in the targets it regulates, and not as much in the targets that are not changing upon knockdown. So kind of widening this out to all the ENCODE RBPs for which we have RNA-seq and eClip data, just showing the same data in a different way as relative enrichment, enrichment in blue versus depletion in red. DDX55 really stands out as particularly significant compared to all the other RBPs in ENCODE where we have this direct data to compare. So the enrichment is very noteworthy. DDX55 looks very interesting for us to follow up. So just a little bit of a flavor of the other follow-up work we're doing. What are the consequences of this potential regulation? Remember, I told you that lengthening or shortening of UTRs can influence these downstream RNA processing events, namely stability. So if we look at sequencing data that measures the endogenous mRNA half-lives in various cell types, these are things like pulse chase type experiments, and we look at the DDX55 targets versus non-targets, confident non-changing, we see that DDX55 functional targets are much more stable, significantly more stable than non-targets. And this is true in both our K562 DDX55 targets and our HepG2 targets. And this difference is highly significant compared to all other RBPs in the two cell types. So ongoing experimental validations are being performed currently to establish if DDX55 knockdown in this context does actually alter stability of its targets, which are in wild type cells very stable. Why might this matter in physiological conditions? Recall that I mentioned in the introduction that alternative polyadenylation is implicated in a number of cancers where altered expression of CPA machinery influences pass choice, leading to largely 3' UTR shortening. So if we apply our kind of same analysis here from ENCODE, what we learn about the regulatory landscape with different cancer data sets, we can really recapitulate the previous analysis from the DePars group of pan-cancer APA shifts, where if we merge our ENCODE data with functional targets from pan-cancer APA events, we see the core CPA machinery that are available in ENCODE being, the most significantly enriched in the overlap of cell line regulation with cancer dysregulation. So we wanted to go a little bit further and look at specific cancers. That previous analysis from the literature is pan-cancer analysis. So looking at some specific cancers, we wanted to see if DDX55 looked interesting in any of them. I'm just showing one of those results. In fact, the most significant association between any RBP and all the different cancer types that we looked at was DDX55 with ovarian cancer. So we identified events that were specifically dysregulated in ovarian cancer, and many of those targets are DDX55 targets. And if we look at DDX55 transcript expression in the ovarian cancer samples, we see that tumors show decreased expression of DDX55, consistent with the large amount of dysregulated targets. So lastly, we looked again at combining binding data with these functional ovarian cancer targets. And I'm showing the meta plot here, much like the ENCODE analysis. Now we're just focusing on our events that are shortened in ovarian cancer, and the DDX55 binding profile looks very reminiscent of what we saw in ENCODE. So we're following up and trying to validate these in an ovarian cancer cell line, and that's ongoing work. So just really quick at the end, all of this has been focused on tandem 3' UTRs, so choosing different poly A sites within the terminal exon. But there are other types of alternative polyadenylation, namely alternative last exons that are influenced by alternative splicing. Myself and others in the Barash lab have worked on Majik version 2, which is our splicing quantification algorithm. And I just wanted to do a little plug there. Now we can identify alternative splicing event types in our complex RNA-seq data. We find that many AS modules contain alternative last exon splicing type events in our ENCODE RBP knockdowns. And if we rank these relative, DDX55 comes out towards the top of this list as influencing primarily alternative last exon choice. So just to wrap up, I showed you our computational pipeline to highlight known and novel regulators of 3' end formation. We've identified a number of putative novel regulators of 3' UTR isoform diversity, including DDX55. And currently we're doing experimental follow-ups to further define the global landscape of APA shifts upon DDX55 knockdown through targeted 3' end sequencing, and also investigating the mechanisms and consequences of this regulation. So with that, I'd like to acknowledge all members of the Barash and Lynch Lab, my funding sources, and you for your attention. But the message doesn't necessarily end here. I encourage you to check out our other related works here at ISMB listed here. And I thank you for your time.